Welcome to lab number 9 on mitosis. Let me share the screen. Mitosis and cell division. We've got a lecture on it in class, and you should know about meiosis and mitosis. And the reading in lab 9 is where to start first. It talks a little bit about mitosis and fission that occurs in prokaryotic cells. So I'm not going to walk through everything here. We've already looked at the word haploid, half the number of chromosomes in the gametes. So there's some background using some terminology that is important to know. This should be complementary information that is very similar to what you've seen or the same as what you've already seen or are going to see in the lecture on mitosis and meiosis. So please read through this, look at the definitions and the vocabulary words carefully. Here we're talking about something that refers to the phenotype and genotype of cells. So first is the genotype. What genes do you have in the chromosomes, the alleles in particular that you receive from mom and dad for a given gene? So the alleles are the versions of those genes that you received. And so, for example, if we're looking at some plants, there might be flower color gene, and capital R could stand for the allele that gives red color, and lowercase r the allele for the white flower. Capital T for tall, small t for short, capital B for branched, small b for a single stemmed branching. Again, these are just examples, and they always use the same letter for clarity. And the dominant trait, the trait that pushes through to the phenotype, which we'll look at in a moment, is the one that is granted the capital letter designation, and the lowercase letter is for the other, or what's called the recessive allele. So with flower color, as an example, if it is dominant for red, if it has one red allele from mom or dad, the flowers are red. Doesn't matter what the other one is because it's recessive. It only takes one red allele to make red flowers. Two red alleles, you still get red flowers. Two white alleles, lowercase r, and you get white flowers. Now, we're not talking about incomplete dominance, but there is something called incomplete dominance where the dominant will show through, but only partially. So for example, with some flowers, if you have one big R from one parent, a little r from the other parent, instead of getting red flowers, you get a pale red or pink flower. Because you are getting the red expression, you're just not getting enough of it to have the deep red color. And so they call that incomplete dominance when you have a difference in the phenotype when you see the heterozygous condition. So what am I talking about there? The phenotype. That's what it physically looks like. So the genotype is what alleles are in the nucleus. The phenotype is what you see or measure. So in the case of red flowers, red is the phenotype. White is the other phenotype. Or in the case of incomplete dominance, you can have red if you have two red alleles. You can have white if you have two white alleles, and you have pink if it's one red and one white. That would be incomplete dominance with three phenotypes. And there are lots of complications in how genes work together, and some are very, very simple, some flower color genes. Most of what we call the Mendelian traits, the ones that Gregor Mendel worked on, the father of genetics, who was an Augustinian monk who developed all of the foundation work for how our hereditary information is passed on, way before we even knew DNA was responsible for it. But he had this idea that you are getting one piece of information from mom and one piece of information from dad somehow, and they were coming through in the children as phenotypes, what you actually see. So, before I went gray, my phenotype was brown hair. I got brown eyes. I got some freckles. Those are all phenotypes, things you can see and measure. So then we get into a few more words here, vocabulary words that talk about the heterozygous versus homozygous. Heterozygous meaning the two copies of a single gene, one from mom and one from dad. The alleles are different. Different. Heterozygous. If they're both the same, big R, for example, you are homozygous for that particular trait or phenotype or gene. So then it goes on to talk about mitosis and the phases. I'm not going to go through that again, but hopefully these diagrams will be relatively simple to follow and complement the in-class or online lecture material. So if you are doing this in class, you will obtain a slide of onion root tip mitosis, and that slide looks like this. And it's not going to be pre-labeled. This is certainly something that's not going to be there, but this is an extremely thin slice near the tip of an onion behind the apical meristem. If I had a larger picture, the apical meristem would be off the slide or off the view of this image below here some distance, but not that far. This is a place of rapid cell division in the zone of elongation. And so we can look at this under a microscope with it properly stained to show the chromosomes, and you can see the phases of mitosis as that root is growing rapidly. Now, because it's a thin slice, some of the cells, you might slice the cell and slice out a piece of the cell. These slices are so thin that if we look here, if you look where my cursor is going down, almost in the center of the image, you can see that there's virtually no detail there at all. There's just one little spot here to the left of this prophase cell here, where you can see what appears to be another telophase going on. And the rest of these, they're cut so thin that sometimes you actually lose the nucleus. So what is the procedure? You get a slide and you just pick a region of the slide, and you don't count the best, most obvious, most exciting ones. You just pick a region and start going down the region and trying to identify what you see. And if you clearly cannot tell what it is at all, skip to the next one. So here we have anaphase, where you can see the clear, almost like inverted V's as they're pulling apart to the poles. It's nearing the end of anaphase. They're almost all the way to the other side. Here's another good anaphase. Metaphase. They're lining up along the middle. There's 3 in a row, which is a little unusual to get 3 in a row lined up right along the middle. This one, you could possibly call it metaphase or maybe early anaphase, just barely starting to pull apart. And if you're an expert at this, you've been doing it for a long time, you've been looking for a long time, you get better and better at this. But the idea is you would say, anaphase, telophase, uh, prophase, anaphase, metaphase. Um, Not sure what's going on here. I think these are a couple of recently divided and they're back in interphase. And what we're looking at is likely the chromosomes that haven't completely relaxed or possibly a nucleolus, but I don't think so. And again, I can't do anything here to the row of cells, the vertical column of cells on the image. So I go to the next one, and most of the detail is lost here. This kind of looks like these might be interphase, but clearly I've lost too much information that they've been sliced off. Now, sometimes by rocking the microscope back and forth, the fine focus, I might be able to say, oh, there is some detail there. It's just out of focus right now, so we can't see it. And then just count till you get 20 of them without any bias to try to pick the easy to identify ones or the clearest, darkest ones. And so this is just a segment of a slide, but your job in this exercise, if you had a slide, is to pick at random 20. And so what I like to do is just go right on down a line or two and pick 20 that are in the region of rapid mitosis. And this slide gives you a pretty good idea of what these different phases look like. Interphase is between mitotic divisions, and then the 4 stages of mitosis. Prophase, where they start to combine or condense, and they start to look a little thicker and more like sausages. You can see the thickness of the chromosomes, but they're not organized in any way. And then they line up in metaphase, they start to pull apart in anaphase, and finally they collect at the poles, and that is telophase. All right. Now keep in mind also that this is happening in 3 dimensions. And so sometimes you're looking at a nucleus and it's not putting the right angle towards you, and you might be looking at a cell that is at an oblique angle or something, and it might not look like anaphase. Something like this, perhaps. Maybe this clearly is more anaphase if I came in at a slightly different angle. Now, the nice thing about root tip mitosis is the cells tend to line up in this rapidly growing region of the zone of elongation. They tend to line up very well for the picture of the camera photo, the snapshot in the microscope. And so we don't have as much trouble about looking at some oblique or odd angle at these stages of mitosis, trying to figure out what's going on. So for example, if I was looking at slides of a rapidly healing wound on skin or something, and I was looking at the healing cells as new cells grew in to heal a cut, they would be happening in dimensions that might be a little harder to see than a root tip. So you obtain the slide, you examine it, you compare the cells. Once you're comfortable, you're going to identify 20 cells. Do not choose the phases, or this will bias your data. You're trying to randomly pick the cells and just read them on down the line and try to give yourself a real indication of those that are identifiable. What is the approximate ratios or percentages or numbers of the different phases of mitosis or between mitosis in interphase? So you're keeping a record of this, and you're supposed to fill out this table. How many did you see out of your 20 in interphase? How many in prophase, metaphase, anaphase, and telophase? And the easiest way to do this is to have one person looking through the microscope, and you just call them off for 10. And the other person in your lab group just checks them off until you get to 10, and then you trade places, or maybe you just, if one person's really good at it, you do all 20. Then the percentage of time. Let's just say, for example, out of 20 cells, 10 were in interphase. 10 out of 20 is 50% of the total, right? 50%. So your percentage time should be the same as the percentage of number. So if you found 2 in interphase, it would be 2 over 20, or 1/10, or 10%. And if it's some other number that needs, to be calculated to get a little better number, like 3 out of 20, which has some decimals after it, calculate is 3 divided by 20 times 100%. Okay? 17 divided by 20. Now, you shouldn't find 17 in one of these categories. Nothing is that dominant. But you might see very high numbers in some, and even a 0 in one of these. That's not uncommon. Maybe you get 1, 2, 3, or 4 in a category. Maybe you get 0 in a category. That's okay, because every single team member in every group in the entire class is doing their own data, and so you'll get class totals. So let's look at an example of class data. So this is just made up. Well, actually, the class data is not made up. The class data was from another class a couple of years ago, but here's some made up sample group data. So maybe in your group, you got 8, 6, 2, 3, and 1 for interphase, prophase, metaphase, anaphase, telophase. And then the class got 81, 57, 39, 36, 27. There were a total of 240 cells observed, 12 groups of 20 cells each. So 24 students put this data together. And so if you want to know the time, telophase was just 27 divided by 240 times 100%. In your group, if you were this group in this mythical fantasy made up data, it would be 1 out of 20, or 5%. 1 divided by 20 is 0.05 times 100% is 5%. Okay? So then this document, which is available to you, explains about how many are in mitosis. Everything in the 4 phases of mitosis is in mitosis. Prophase, metaphase, anaphase, telophase, they add up to 159. The only thing you don't add up is the interphase. Oops, and there's a typo here. It should say, and I'll have to correct that, it should say 240 divided by 81 is 159. So that was a typo, my apologies. And I will re-upload that file in a moment. But either way, the answer is 159. And so the mitotic index is how many cells are in mitosis? 159 out of the 240. At any given time, these rapidly dividing cells are in mitosis about 66% of the time, based on this particular class data. Now, if you assume that there is some sort of a cell cycle that we've measured separately. You can't measure how fast cells are dividing on a dead slide. This would have to be done using other methods on live tissue. But if somebody tells you that the cell cycle in a root tip of rapidly growing onion roots is 16 hours, well, then you could say it's in mitosis 66.25% of 16 hours, or about 10.6 hours in mitosis. And the remaining, what would that be? The remaining 5.4 hours would be in interphase, doing things like growing and DNA replication, getting ready to divide again. That would take about 5.4 hours based on these data. Okay, so that's just a sample of the data. And then you have some questions. The mitotic index, I just explained that to you in that diagram. The number of cells in mitosis, everything except interphase, divided by the total. And so for this calculation, you can repeat or redo the data that I have in the sample and just show me you know how to write what I just demonstrated, essentially copying the information. Or you can also do the mitotic, excuse me, no, I gave you the group data. You're going to do that with the made up data. Now, if you have a lab partner that actually did it in class, or you have other access to class data, then use that. So the mitotic index based on your slide would be this calculation. You're going to add up all of the mitosis phases divided by 20, leaving out interphase. And then the B is what I just showed you. I've already given you the answer to B, unless you're doing it for real class data and you're doing this lab online just because you missed that day, but you were able to get the class data and group data from your partner or from me. Are you more confident in the accuracy of your percentage calculations based on your data or the class data? Which one do you think is better, a better estimate of the actual mitotic index? The cell cycle of the onion root tip lasts for about 16 hours, as we just learned. How long in terms of hours and minutes does mitosis last in an onion root tip cell? So the entire cell cycle is 16 hours. How long does this last? Just the mitosis minus the interphase. Okay, so again, you're going to calculate the time for mitosis based on your data. How much time is it here in your personal data, personal group data, versus the entire class data? How much is it? Now, I've already given you how to do that for the entire class data, but unless you get the class data from the current class that you're in, if there is a face-to-face component, and you can get a hold of that data from your lab partner or from me. All right. And how are mitosis and meiosis similar? How are they different? This was a question we got into. What are the two main differences? The two biggies from lecture. Please look at the lecture on mitosis and meiosis. Other than root tips, where might you expect mitosis to occur in plants? Again, there's lots of examples that you could use. Give me a few. Why is mitosis inadequate for producing sex cells? Again, that should come from the lecture portion. And how many, or excuse me, number 8, many asexually reproducing organisms reproduce by simple mitosis. What is one source of genetic variation for these populations? So if there are asexually reproducing eukaryotic cells and they just go through mitosis, how can there be variation? What could be a source of variation in their population? Think about that. I'm not going to just give you the answer. If we've looked hard enough at different organisms, even the ones that generally reproduce asexually and do that almost all the time, you look long enough and hard enough and with enough scientific effort put at it, you eventually find that there is a sexual cycle somewhere, sometimes. I don't know of anything that has been proved to have no sexual life cycle of a eukaryote. And that's it. Lab number 9.